Well, good afternoon. My name is Lee, and I come from the Tel Aviv University in Israel. And today I'll be presenting a study we conducted on signal transmission via tobacco plants for the applications of body channel communication. We conducted this work in collaboration with people in the plant biology department at the Tel Aviv University, in addition to our collaboration at the Politecnico de Torino with Professor Danilo De Marchi's group. So an outline for my talk will be, I'll start with plant body channel communication. I'll explain our experimental setup. I will show the results of an electrode study that we conducted, and after that we started a linearity study of the plant medium, and eventually I will summarize. So, as we all know, body channel communication is actually used to transmit the signals across the human body, having the human body itself as the transmission medium. The actual transmitters and receivers, or effectively our electrodes, are connected directly to the human body. That means they can be connected in contact with the skin in a capacitive manner, or they can be implanted into the body or inserted in an in vivo manner. It's known that body channel communication is an effective way for creating a wireless network of different sensors. It allows the support of low power devices, and it eliminates the need for external wired communication devices or pieces. So here what we introduce is plant-based body channel communication. So plant body channel communication actually means that we're using the plant medium as the medium for the signal transmission. And this approach was only quite recently suggested and published by Professor De Marchi's group in collaboration with the Tel Aviv University. So what we're suggesting is that the plant body channel communication could be used for plant Internet of Things and for systems and integration in plant biosensors. So here what we present is some early studies on this approach. So our experimental setup consists of a tobacco plants. We use young tobacco plants for our experiments. They've been grown at the plant biology labs at Tel Aviv University. And they will usually be grown for a few months, between say 2 to 3 months. We couple our electrodes in a galvanic manner. We use gold wire electrodes, and we couple them directly to the stem of the plant. And while we're assuming that that signal conduction will be via internal tissue within the plant stem. So as you can see in the schematic description here, the electrodes are coupled along the stem at different distances. So the distance between our transmitter and receiver is approximately 5 centimeters. The actuation and data acquisition are done using a single generator and an adjuvant oscilloscope. So what we wanted to study was the actual electrode configuration, which has been shown in human body channel communication to affect the signal attenuation. So in order to check these, we connected our signal and ground in different configurations. So if you look at the diagram over here, you can see that we've denoted G and S, to G being the ground, S being the signal. And we have altered their positions in the different configuration, calling them GSSG, SGSG, and comparing the attenuation from these different configurations. In order to do this, we inserted a rectangular pulse with on-off keying. And we looked at the change across frequencies when we measured from 10 Hertz up to 100 Hertz. No, sorry, up to 10 kilohertz. Sorry. And what you can see is that the attenuation trend is very similar to that shown for human body channel communication. You can see that there is no real preferable configuration. If we look, we completed these measurements across a few days and a few plants. And here we've given an example of 2 different days, not consecutive, first day and third day. And we can see that the relation between the different configurations across time does change, yet there isn't a preferable one. And then if we look at the mean swing across the frequencies, it gives an attenuation of about 3.5 dBs. So what we can say is that we can't say for certainly that there is a preferable configuration, that there would be a best choice from these observations. After we did this, we also wanted to see if we could determine whether the plant stem behaves as a linear system. So I would like to know if it's an LTI system, okay, linear time invariant. So in order to define an LTI system, we need to look at signal transmission via the stem. And this was done using rectangular pulses with a duty cycle of 20% and frequencies ranging again from 100 Hertz to 100 kilohertz. The maximum voltage inserted here is 500 millivolts. We have reason to believe that going above 1 volt may have an effect on the hydration of the plant, and it may harm the plant tissue. But so we've kept all our measurements under this value. So if you have a look here at the graphs below, you can actually see the output voltage as a function of time for different inserted pulse widths, starting from 2 microseconds and reaching 400 milliseconds. And just from a quick observation, you can see that we have a few time constants here. So that indicates we have some kind of capacitive behaviors within the plant stem, and they behave slightly differently. So next, what we wanted to look at is to see the impulse response and study the fall times and to sort of understand what nonlinear conduction mechanisms we have. So we looked at the impulse response and its derivative. So if we have a look at the graphs here, you can see the fall time as a function of the impulse, the inserted impulse voltage. And you can see quite clearly that the fall time is dependent on the inserted voltage. which means that we have some kind of voltage dependent load. And it needs to be further studied. So overall, these are the experimental results that we've obtained so far. And we can say that single attenuation in body channel communication consists with the trends shown in human body channel communication. We cannot say that our study on electrode configuration shows a preferable ordering of the electrodes. We can say that plant stem does exhibit certain non-linear behaviors under specific conditions. But what is for sure is that we have to conduct further studies in order to really further establish this approach. So thank you very much for your time. And I guess now it's time for questions.